afternoon. Good afternoon, everyone. So the title of my presentation is Food Effects on the Gastrointestinal Transit Properties of Amphotericin B, prepared in solid lipid nanoparticles. We've heard a lot about nanoparticles this morning, and particularly about amphotericin B. I was quite fascinated on the talk just before lunch about amphotericin B. So what we've done here is to look at the food effects on the gastrointestinal transit properties of amphotericin B. But before I go into the details of the work, just a brief introduction that this drug is very effective, as we've heard this morning, in treating very serious fungal infections. However, it's currently given by intravenous route, and it comes with very serious side effects, as we've come to know. And research has shown that this drug can have slightly lesser side effect when it's given by the oral route. However, it's very poorly soluble, as we've heard this morning as well. So there is an interest to develop this drug into alternative delivery systems, which can improve the bioavailability. And so we are interested in this area as well. But we are aware that no work has been carried out to see the transit properties of amphotericin B dosage forms, and particularly to see which part of the gastrointestinal tract this drug is best absorbed, if the oral route is to be chosen. So the purpose of the work was to formulate amphotericin B in solid lipid nanoparticles intended for oral delivery, and to study the gastrointestinal transit properties of the dosage form, that is the solid lipid nanoparticles, but also to see where exactly in the GIT amphotericin B is best absorbed, and also to see the effect of food as well. So whether food has any effect on the absorption of amphotericin B within the gastrointestinal tract. We decided to use a very not so sophisticated method, but it's nonetheless acceptable. So we use marker drugs to study the gastric emptying properties, as well as the cecal arrival of the dosage form. So amphotericin B was prepared in the same way as paracetamol nanoparticles, and the paracetamol, as you are all aware, you might be aware, is very rapidly absorbed from the small intestine. So it can be used as a means of determining the gastric emptying, or the arrival of the dosage form in the small intestine. Sulfasalazine can be hydrolyzed into sulfapyridine in the large intestine and be absorbed, and therefore it can also be used as an indicator of the arrival of dosage forms containing this drug at the cecum. So these were prepared and were characterized with regard to size, charge, morphology, among other things, and were studied on animals. So we used male Sprague Dowley rats weighing between 250 grams. And of course, the work had an ethical clearance, and the animals were divided into two groups. So there was a fasted group and the fed group. And each animal was administered a single dose containing the 3 drugs in the nanoparticle delivery system that had been dispersed in deionized water, and blood samples were taken from the tail at predetermined time intervals. In terms of the gastric transit parameters, the gastric transit time, or GTT for short, of amphotericin B was measured as using the plasma concentrations of the paracetamol. And that's, as I said before, is because paracetamol is very effectively absorbed from the small intestine, and that can be used as a measure of the arrival of the nanoparticles in the small intestine, or gastric emptying. So that time was taken as a time for 10% absorption, because the nanoparticles are a dispersed system, and it's a continuous system. So the arrival is not a one event, it's a continuous event, just like pellets. So 10% absorption of paracetamol was taken as the time for arrival of the nanoparticles in the small intestine. And the time for complete emptying of the nanoparticles from the stomach was taken as a time for 90% absorption of paracetamol. That was the gastric transit time. That is the time that the solid lipid nanoparticles remained in the stomach. The small intestinal transit time, which is the time that the dosage form spends in the small intestine, was taken as a difference between the T10S, which is the time for 10% absorption of sulfapyridine in the large intestine. So that was, we subtracted the T10P, which is the time for 10% of paracetamol absorbed, from the time for 10% of sulfapyridine absorbed in the cecum. Okay, sorry for the lots of, but I need to define these parameters before I go into the details. So the cecal arrival time, I said, is the time when the dosage form appears or arrives at the cecum. And it has been measured in some research work as the time when the sulfapyridine is detected initially in the blood. But this could lead to an underestimation, sorry, it could lead to an overestimation of the time because of the fact that sometimes you have the drug being released from the dosage form, so that could be detected rather than the dosage form actually appearing or reaching the cecum. So we decided to use the cecal arrival time as the time for 10% of sulfapyridine absorbed. That we felt was much more realistic. The colon transit time was a time for 90% of sulfapyridine absorbed, or T90S. So the idea was to try and produce these nanoparticles such that they had the same or similar characteristics. So they were characterized in terms of their physical properties. And the graph on the left, the bar chart, is showing the zeta average. So these were statistically not different. So the paracetamol, amphotericin B, and sulfasalazine nanoparticles were quite similar in terms of their size. Also similar in terms of their morphology from the SEM image. They look quite similar and well dispersed. And the zeta potential were quite comparable as well and were negative. We heard this morning how negative zeta potential can be relevant. These were negative as well. So we did a drug release study, and if you can see in all the cases, the paracetamol had the highest release, in vitro release, followed by sulfasalazine and amphotericin B. This is actually expected because paracetamol is very soluble in water compared to sulfasalazine, and sulfasalazine is also well soluble in water compared to amphotericin B. The graph is actually showing the counterpart for the freshly prepared as well as a freeze-dried counterpart, and there wasn't much difference between the two pairs, as you can see. We also tried to see how the particles behave when they are subjected to some kind of constraint. So we exposed the nanoparticles to agar. That was sort of to see how the nanoparticles would behave in the presence of food. I guess there could be some other way of doing this, but this was nonetheless quite relevant. So we exposed the nanoparticles in an agar gel, and then we applied some potential to see how they will migrate when they exposed to this charge. And you can see in the right panel that the extent of traveling as well as the rates was quite similar in all three cases. We also tested the viscosity of the nanoparticles, and all of them were essentially quite similar. So this gave us the belief that we can use the profiles that we have from the pharmacokinetic parameters to give an indication of how all the nanoparticles will behave in vivo. So this is a key assumption, is that since all the particles are quite similar in terms of their physical characteristics, we believe that we could use the behavior of one of them to infer how the others will behave as well. So it's a very key assumption. We believe that is the case. These are actually graphs showing the effect of food on the absorption of paracetamol. The left graph is showing the profiles for the fasted and the fed states. And the one on the right, the one on the left is paracetamol, and the one on the right is amphotericin B. I think the key thing I want to emphasize here is that in the case of paracetamol, you can see that there is a peak right about here, no pointer. I hope you can bear with me. So there's this peak over here, which is about 8 hours in the fasted rats and paracetamol administered rats. And we see a similar peak as well in the case of the amphotericin B, also about the same time. And we believe this is because of the slow process. Since these are lipids, they are taken up by the pious patches and through the lymphatic system and empty into the blood much later. That's why this appears late, and it seems to be irrespective of the drug load. So whether it is amphotericin B or paracetamol, we find that this is happening, and especially in the fasted state. So it gives that peak much later because of the slow process, the slow uptake process of amphotericin B, and that's why that happens. In the fed cases, however, you find that the profile is quite flattened, especially in the case of amphotericin B, it's much flattened. Food tends to sort of slow the rate of absorption. Again, this is something that is expected, and something similar is observed in the paracetamol profile as well. So if we put aside the profiles for amphotericin B in the fasted state and the fed state, against the absorption from the control, which was just a suspension of pure amphotericin B in deionized water, we find that in both cases there is a significantly higher absorption from the amphotericin B compared to the suspension. So in the fastest case, that's what we see over here, and so in the fed case, again, is higher. But statistically, we observed that there was only a significant difference statistically in the case of the Cmax, as you can see over here. Yep. And the AUC was comparable in the case of the fed and fasted state. Yep. The Tmax obviously was delayed because of the presence of food. Again, this is something that is expected. If we compare between the control and the amphotericin B solid lipid nanoparticles in the fasted state and the fed state, so separately, again, we find something interesting. In the case of the amphotericin B solid lipid nanoparticles, we find that there is a delay in the Tmax with the nanoparticles compared to the control. So it's 8 hours plus in the case of the amphotericin B nanoparticles, and it's just 1.5 hours in the case of the control. And the Cmax is about twice the case of the amphotericin B nanoparticles, and it's just about 2 times again in the case of the AUC. And we see a similar result in the case of the fed state, but the fed state seems to have a slightly better profile in terms of the, if we compare with the controls, I'll just take them separately to reduce the confusion. If we compare the amphotericin B solid lipid nanoparticles in the fed state, again, we find that it's highest in the case of the nanoparticles compared to the control. Yep. So similarly to the fasted, we find the same situation over here. So we try to compare the transit times, that is the times that the dosage form spends in the different sections of the GIT, in both the fasted state and the fed state. And you can see in all the cases here, if we compare this second column with the first column in this first table over here, you find that there is an increase in the transit time in the fed state, also an increase in the small intestinal transit time in the fed state, and the same is the case with the cecal arrival time and the cecal transit time. All of them seems to be increased when there is food in the stomach. Again, this is something that is expected. Obviously, CTT is most significant in all the cases. And we try then to look at the percentage absorption in the different sections of the GIT, starting from the stomach, small intestine, and colon. First, in the fasted state, we see again that we have 44% absorbed in the intestine, just 2.8% in the stomach, and 53% in the colon. We compare that with the fed state, we find a similar profile, a slightly smaller percentage absorbed in the small intestine, but a much higher percentage absorbed in the colon in the fed state. The reason why we have this colon plus here is a suggestion that the absorption of the solid lipid nanoparticles, or the uptake of the solid lipid nanoparticles, is occurring while the dosage forms would have reached the colon, but also it's likely for some absorption to be taking place through the lymphatic system, which is a slow process anyway. So it's a combination of the uptake process while the dosage form was in the small intestine, while it goes through the lymphatic uptake, plus what would be happening in the colon itself. That's what's given by the plus. And on the basis of this, we believe that obviously this is just summarizing some of the key points, that we believe that since the food has delayed the transit of the dosage forms in the gastrointestinal tract, it appears that there is better absorption. That is to say that the uptake process is very slow, and as a result of that, its emptying into the lymph is much delayed. And therefore, we believe if we can develop delivery systems to slow down this process of transit within the GIT, we are likely to have a better absorption as well. So in conclusion, we can say that we've been able to successfully produce these nanoparticles in solid lipid form, and it's certainly much more better in terms of its bioavailability compared to the control in both the fasted and fed state. We've also seen that food doesn't seem to affect the bioavailability that significantly, and the absorption process in the small intestine appears to continue even after the dosage form has reached the colon. So this is a key point that we've observed in this study, and therefore further work to be done, or further work done on delivery systems containing Amphotericin B aimed for oral delivery should be made to have a slow transit. If this transit is slow, then it's likely that the absorption will be much more enhanced. And actually, we're working on this at the moment. Thank you very much.
This is certificate of recognition.
Thank you.
Great work. Continue the good work.
Thank you.
Thank you very much. There is an announcement.